Thank you, John. So I will be sharing my screen shortly, but before I start, I'd like to congratulate you on the new directorship. This is going to be a great future for the institute moving forward, as well as I noticed Peter Bradshaw is on the line as well. So thank you to your continued support to the institution. This allows researchers such as the panelists today to continue doing and having fun in the lab, and also potentially changing the world and the mining industry while we're having fun. So I'll start my screen. So hopefully you should be able to see it, and I'll go to the full screen mode. All right. So one of the big challenges with Zoom that I have noticed, especially when I've been taking lectures, is you can't really read the audience. So if this statement that I say falls flat, I do not take any of the blame. I will ascribe that entirely to Zoom. So some messes in the world, they cannot be cleaned. Okay, and the Canadians in the audience are probably chuckling when they hear that. But then there are some other messes in the world that are tailor-made for synthetic biology. And one of these things that I would like to talk about is the treatment of acid mine drainage. And I would like to convey to you how this emerging domain at the interface of engineering and biology, which we term synthetic biology, is transforming the way we design and develop processes for extraction of raw materials, natural resources, as well as extending those to the remediation of the environment. And this resonates with the mining industry, just given the span of applications that synthetic biology actually has in this industry. So before I start, and you probably have got a flavor of what BRIMM and especially the subgroup on the mining microbiome is doing, is essentially applying biotechnology to address pinch points in the entire mining value chain. And this starts all the way from extraction processes. So as John had mentioned, Jetty was in the news lately, and they are working extensively on hydromet as well as biohydrometallurgy to improve extraction processes. And this is some place where biology as well as synthetic biology are having tremendous applications. And my research group actually has some work in the space of extraction as well. And then moving forward, as you start adding value to these products, there is processing. And the talk for today will address a project that we're working on in the lab that addresses aspects of processing as well as what comes thereafter, which is waste management. And of course, as Stephen and Rachel had pointed out, there are enabling technologies that are allowing synthetic biologists and engineers to design and develop applications in each of these domains. And that is, and one of those big things is, so not only are you establishing the technology platforms for each of these, but also through barcoding and other types of applications, you're extending this to environmental monitoring. But the talk for today, which is quite brief, and I'll actually give you a big picture overview of what we're doing in the domain of waste management. And you could actually be generous and extend the boxes of this particular project all the way to processing as well. And before I describe specifically what you're doing, I guess it is fitting that we describe what the nature of the challenge is that we're trying to address in the industry. So if you were to take a typical open pit mine, now there are obviously variations, but this is just based off information that if you go and calculate some of these statistics for these mines, on average, a typical open pit mine, now you could be plus or minus these numbers, but let's assume that this mine is producing 3 million tons of ore per year. So we start right at the base of that diagram. As we realize, mining and processing are extremely energy and water intensive processes. So some of these processes and some of these mines, they end up utilizing as much as, so if for 3 million tons of ore per year, you're looking at approximately 4,000 gallons per ton of ore mined. So that translates to about 12 billion gallons of water that are being used. And such is this volume of water that has to be treated. They're essentially stored in these ponds and tailings ponds. Now, as these tailing ponds are sitting in the environment, you're having sulfides, you're having oxygen, you're having bacteria in these environments, as was described by in the presentations before. This catalyzes some very interesting chemistries, and that leads to the formation of acid mine drainage. So you're having roughly about 238 million gallons per year for a typical mine. That volume of AMD or acid mine drainage is being produced. And this is extremely corrosive. It is a danger to the environment, and therefore it needs to be addressed. And the way acid mine drainage is addressed is to use quicklime to neutralize it. So you would then, for these types of volumes, you're looking at approximately 4,500 tons of quicklime. It has to be of a certain specification in order for you to use it. And that results in a total bill of about, a bill of goods of about $2 million per year. Now, the CO2 emissions that I have put here, they don't, and this is a very, very simplistic calculation that refers only to the production of lime. So every ton of mine produces about 0.8 tons of CO2. So that is just the CO2 that is being emitted from the production of lime. This does not even factor in some of the CO2 that's coming from some of the other operations, which is quite sizable. So how is it that synthetic biology is actually trying to address this? So what we're trying to do is to treat that number at the top, 238 million gallons per year, and try and come up with a better solution to that. And of course, try and eliminate some of this heavy mine lime utilization at these sites. So in that, as you add these numbers up, it translates to quite a sizable cost savings as well as quite a sizable reduction in CO2 emissions. Something that is also related to that is once you produce the sludge has to be disposed. The disposal cost for those volumes is roughly about an additional $500,000 per year. And that does not even factor in some of the logistical challenges that are present in that process. And then there is the other issue of regulatory concerns. So even though this is sludge, and this has been sitting as a waste stream or a byproduct stream, it is still teeming with a lot of valuable metals in there. And that represents both a concern for discharge into the environment as well as lost value. And the total concentration of all of these metals is roughly, you add them up, obviously some metals are more than others, but you're looking at about 3 grams per liter. So what is it that we have been trying to do? So in order to address this challenge, building off of the excellent fundamental as well as discovery-oriented research that collaborators such as Stephen Hallam, Sean Crow, Rachel have spoken about, Scott Dunbar as well. I have the distinct privilege of working with all of these panelists. We are essentially building off of that excellent fundamental knowledge to then try and apply it. So my view of the world is, when Stephen sees a rose, he starts admiring the beauty. And when I see a rose, I essentially ask how much perfume I can extract from it. And that's just the engineering-centric view of the world. So we are using a combination of 2 processes, a microbial fuel cell in combination with another process called microbial-induced calcium precipitation. And by synergistically applying these 2 processes in that entire workflow, we are able to address all of those challenges that we are talking about. We are trying to reduce lime usage. We are trying to recycle more of the lime. We are recovering some of the lost value of the metals in those streams, which is not only a money earner, but it also helps address some of the regulatory challenges. And then, of course, you are eliminating or reducing the production of sludge. So you're saving you're saving disposal costs as well as material handling costs. So the way we're describing this is that it is both a value addition, you're saving costs as well as you are reducing expenses. So how do these 2 processes work? So research in my lab, and this is a collaboration with Scott Dunbar and his team, is we have been working quite extensively on designing and developing microbial fuel cells. So microbial fuel cells are essentially they are electrochemical cells. The big difference is that the the oxidation reaction, the reaction that generates the electrons that allow for the metals to get deposited and pulled out from these wastes, that reaction is catalyzed by a special type of bacteria. They're called exoelectrogenic bacteria. So these bacteria, they consume an organic substrate, they oxidize it, produce the electrons that are then going all the way to the cathode where your metals are being deposited. Now, this process generates hydrogen and CO2. And what we figured out is you can also loop this process with some sort of a carbon fixation process. So organisms such as Clostridium are able to fix the carbon dioxide and produce the organic substrate. In this instance, it would be acetate or lactate. And then the organism in the chamber that is actually consuming the substrate is usually an organism known as Chivinella onidensis or Geobacter sulfurreducens. These are the bacteria that are then consuming the substrate and then depositing the metal. So that is the general principle of the microbial fuel cell. Our specific research is aimed at, first of all, looking at the construction of the cell, and then engineering the bacteria using some of these information, the biological, biochemical information that my colleagues are identifying, and try and engineer, genetically engineer a more productive microorganism. So among other engineering that we're looking at is we're looking at the conductivity of the surfaces of the cells, and we have done some extensive research on this field. So as we speak, we are able to actually deposit about 90% of copper in about, which is roughly about 250 milligrams per liter in about 6 days. This is obviously bench scale research, and the cells are able to generate a power density of about 10 to 20 milliwatts per meter square. So this is definitely among the higher numbers that have been published. And really what we are now doing is taking this towards larger scales. And as you are able to scale up these processes, it raises some very compelling images of how you could use microbial fuel cells to address a range of problems at the mine site. In terms of the microbial induced calcium or calcite precipitation, you're using another microorganism. So this is taking place in a second stage. You're using microorganisms such as Sporosarsina pasteuri, and that microorganism is playing off of two enzymes. So urease breaks down urea to produce ammonia and carbon dioxide. Then another enzyme, carbonic anhydrase, which is one of the fastest enzymes known in nature, that is able to then produce carbonic acid, which is then precipitating the calcium ions that are in there to produce calcium carbonate. So so how does the MICP and the MFC actually work in concert? And so before I come to this slide, so the way the process actually is it goes through an MFC cascade where you're recovering a lot of the metals that are in the stream. You use lime to neutralize that, the acidity of that solution. There is a pre-treatment step where you're dissolving the gypsum in the sludge. And then once you have the calcium ions that are in there, you're using MICP to regenerate CaCO3 initially, which is then used to produce lime. And this lime is then looping back into the process. So we are trying to impose some circularity into these processes, which are inherently more sustainable and more efficient compared to your traditional linear processes. And just to show you in terms of what other extensions that we have applied these results to, we have used those same results, namely engineering bacteria to make them more conductive. This is something that we're doing to improve the performance of the fuel cells. We use that same technology to build solar cells from bacteria. So this was something that was in the news recently, and this builds off of the same foundational information that we're using in the microbial fuel cells and the MICP. So with that, as I say, the salient benefits is you're reducing waste, you're eliminating this huge tonnage of sludge, you're recovering value through the deposition of these metals, and then ultimately it translates to saved costs in terms of reducing the lime, as well as more earnings through commercialization of some of these recovered metals. So ultimately, we bring a lot of technologies to bear in order to achieve these things. And a lot of these specific domains have been discussed by some of the previous panelists and will be discussed by Susan after me as well. But ultimately, we view ourselves as the aggregators. We take all of this incredible information that is within the BRIMM ecosystem, we connect it to a very clear pinch point in terms of an industrial process, and we then try and apply these things in order to develop greener processes, as is the case over here, as well as better products, more energy efficient products. This is my team. They're an incredible bunch of talented individuals. Some of them are in the audience, and none of this would be possible without their enthusiasm and their energy. And what we like to do is we like to feed off of their ideas. So with that, I will conclude. Thank you for your attention. Thank you, John, for providing me the opportunity to speak. And over to you, Susan.